And I would like to thank the organizers and GCRF for the opportunity to speak. I have to say I would love to take part in this bike ride in South Africa, as my colleagues who sit in the back and are part of this work know I'm an avid biker, so it's really, that's nice. Can everybody hear me in the back? Good. So, as I said, I work in Peter Besmer's lab, and in Peter Besmer's lab, we try to model this disease that some of you are suffering from in the mice. And in my presentation today, if it's working, let's start. I want to focus on two little questions, which are very basic in the end. What is the molecular basis of GIST? And how can we prove that? What is causative for GIST? And how can imatinib resistance occur? As I said, the approach we take in Peter Besmer's lab is to model GIST in mice by introducing the mutations that we find in human patients in the tumors into the mouse germlines, meaning the mice carry the same point mutations in their genome, and then we see if GIST develops. A little bit of background. So, as most of you know, most of the mutations occur in the receptor tyrosine kinase KIT. And KIT is a transmembrane receptor tyrosine kinase, which means it's a protein that is crossing the membrane, the cell membrane, and it's basically waiting to receive a signal. So here, the signal usually in a normal unmutated setting is KIT ligand, and upon binding of this signal, you facilitate that this receptor turns on and then can transmit into the cell. So this is the outside of the cell, this is the inside, into the cell signals. Overly simplified saying, KIT is on, tyrosine, as it is a tyrosine kinase, is on in an on state, and this is sending the signals to block a programmed cell death and to enhance cell proliferation in a normal wild-type setting. And that's there, it's reversal. If we now have activating mutations in KIT, most of which are in exon 11 in the juxtamembrane domain, as you know, then, overly simplified said, the KIT receptor is stuck in this on position. It doesn't reverse to the off position, and so we get a sustained blockage of programmed cell death. The cells don't die anymore, and we get sustained cell proliferation, both of which are hallmarks of cancer. So a postdoc in the lab before me, Gunhild Sommer, she set out to model GIST by introducing a single amino acid deletion, the deletion of valine 558, into the mouse germline. So the only difference between this mouse and a normal mouse is that you have one single amino acid in KIT missing. And sure enough, this mouse developed GIST. If you do cross-sections through the stomach or the cecum, you might think about it as the appendix of the mouse, or the colon. In these cross-sections, Christina Antonescu, our pathologist, can see that there's GIST development in this cecal region that is morphologically very, very similar to what we see in humans. So if you would present Christina with a cross-section through a human tumor or this mouse tumor, she probably cannot make out the difference. And we also see overproliferation of the progenitor cells for GIST, the presumed progenitors in the stomach and in the colon. So all these cells are hyperplastic, overproliferating cells. And so this mouse model really proved that a single point mutation can cause GIST, at least in mice, and that it is sufficient to initiate this tumorigenesis. So towards this first question, what is the molecular basis of GIST? At least one part of the story is we have single point mutations in KIT, which are sufficient to induce GIST in mice. Most of you might be interested in the story of resistance occurring. So how can imatinib resistance develop? As we have modeled in the mouse before, we introduced an exon 11 mutation into the germline of mice. We now wanted to build up on this knowledge and combine it with second site mutations. Second site mutations are found predominantly in exon 13 or exon 14 or exon 17 of the KIT receptor. And we modeled after this example, as Christina and co-workers found out, this exon 11 mutation combined with this exon 14 mutation, so introducing these two mutations at once into the mouse genome. The second site mutation that we are modeling here is a very specific one, and don't get me wrong, there is this wide spectrum of a lot of different second site mutations, so we are only modeling one of them. And this specific one is in the active site of KIT. So KIT is here in gold. And it's surrounding what is modeled in blue is imatinib. So that is the drug, the inhibitor imatinib here in blue. And as you can see, KIT forms stabilizing interactions with imatinib. And one critical stabilizing interaction with imatinib is made from this residue of KIT, the threonine 670, and when this threonine 670 is mutated to isoleucine, as depicted here, T670 mutated to I, isoleucine is much bulkier. It's basically then poking into this pocket, and you can imagine that this inhibitor, imatinib, as you might know the name Gleevec, the commercial name is Gleevec, cannot fit into the pocket anymore. So, and this is not only happening in KIT cases, but also in other mutations in the PDGF receptor and other receptors as well. So it's a common mode of resistance, and thus we were interested in modeling this common mode of resistance. So we introduced these 2 point mutations into the mouse genome, and I will refer to it as exon 11 plus exon 14 mutant, to make it a little bit easier to follow. And the findings we see in these mice, which are double mutant, are similar to the ones in the single mutant mice. So in single mutant mice, we have overproliferation of these cells, which usually only form a thin layer. So we get expansion of that here and here in the stomach and in the colon, and we see similar findings in these double mutant mice. Most importantly, also the double mutant mice develop these GIST tumors. So per se, it's recapitulating what we saw in the original single mutant mouse model. We recapitulate and see in the double mutant mouse model similar findings, but obviously the question is, how does this mouse with 2 point mutations respond to the standard therapy? Standard therapy, imatinib, or commercial name Gleevec, is depicted here, the molecular structure, and again this connection to this threonine 670 of KIT that is stabilizing it in this pocket where it's inhibiting the KIT action, and a similar interaction is seen for the inhibitor Dasatinib. So we are testing Imatinib and Dasatinib, as well as structurally unrelated compounds Sunitinib and Sorafenik in these mice, one after the other, and want to figure out how do these mice respond to these inhibitors. And how do we read it out if mice respond to these inhibitors? One way is to take a look directly at KIT. Is KIT in the on state? Another way, and biologically a little bit more relevant, is also downstream of KIT, as we say. In which state are the proteins within the cell? Are these proteins in the on state, then enhancing, for example, proliferation, or are they in the off state? And I want to focus your attention on these 2 proteins, S6 and MAPK, short MAP kinase. And when KIT is in the on state, these proteins are also in the on state. So it's a readout of KIT being on. And if you inhibit KIT, the idea is once KIT is inhibited, also these proteins should be in the off state. So we can use S6 and MAP kinase as readouts if our therapy is working in the mice. So we treated mice, and first with imatinib, and then with sunitinib. And in untreated mice, if MAP kinase is on, we can detect this on state with an antibody, and then we get brown staining of cross-sections through this tumor. Also, when S6 is on, same picture, brown staining in the cross-section through the tumor. If we treat with imatinib, we see a rapid downregulation of this on state. So the drug is working in the single mutant mice. If we now test the same drug at the maximum tolerated dose in mice that have two mutations, exon 11 and exon 14 mutation, we don't see any response at all. Basically, these mice look like as if they were untreated. So this mouse model is truly, with this combination of exon 11 and exon 14 mutation, truly resistant to imatinib. But obviously, it's important to see, can we somehow overcome this resistance? So we treated these mice with sunitinib, and there we can restore the response. The initial response we saw with imatinib in the single mutant mice, we can also achieve with sunitinib in the double mutant mice. We also tested Dasatinib and Sorafenik. Dasatinib works on the single mutant mice on the tumors, can shut off the signaling in the tumors. Dasatinib does not work in the double mutant mice, so with this specific configuration. But Sorafenik can overcome the resistance again. So sunitinib and Sorafenik is this close analog of regorafenib, which is in the news right now, so can overcome the resistance. So, and with that, I want to close and summarize that we can actually model the tumorigenesis in mouse models, and showing that a single point mutation in KIT is sufficient to induce GIST development in mice. And then the imatinib resistance we can model as well, just adding on a second mutation into the mouse genome. That's all the difference is between these mice and normal mice, causes resistance to imatinib. And so we have now this mouse model in our hands, and obviously we are thrilled to test more second-generation drugs in this model. With this, I want to acknowledge, obviously this is not possible to do all this alone. A lot of collaborators, most of them sit in the back at the table from the lab. Ferdinand Rossi is a postdoc in the lab, and Gunel Sommer made the single mutant mice. They were very instrumental. Obviously, my mentor, Peter Besmer, who initiated all this work, he's sitting over there, and who actually was the very first one who worked on KIT, because he discovered KIT in the mid-80s, and since then his lab is working on this. And Christina Antonescu, our pathologist, who always provides excellent service to when we need the analysis of these mice, the pathology. And a lot of core facilities here at Sloan Kettering, as well as across the street at Rockefeller University, and funding. And again, I would like to thank for the opportunity to speak today to you. Thank you very much. Any questions? That's a very good question. We are not sure. If the expression level is changed, The tumors are different in size. So we get tumors, but they are different in size in these different mouse models. And so we haven't nailed it down. We don't know if there's, we didn't do like any close examination, say via RT-PCR, and then following up on that in terms of loading. But the phospho-KIT seems to be unchanged. So it's highly phosphorylated. So when KIT, meaning KIT is on. So phospho-KIT means KIT is on. So she was asking about the on state of KIT. And so we see the majority of KIT is in this on state, in the phosphorylated state.
Like, we should show, like, Sunitinib work well in your double mutation mice, especially if they work on the like kinase pathway and also the phospho-base 6 pathway. So have you any
That's a very good question. So the question was basically, I was looking now downstream of KIT. What is happening within the cells? Are these cells in the on state to keep proliferating? And to us, this resistant mouse model is a very good scientific tool now because it is resistant to imatinib. So when we treat with imatinib and nothing happens, that means imatinib in the setting of GIST really is specific for KIT. So meaning if imatinib cannot bind to KIT, and it could still bind to other proteins that could also be involved in this setting, but it doesn't do that. That shows us, at least in this modeled setting, everything is really KIT dependent. So it's really important to inhibit KIT to get a response in this system. Did that answer your question?
I would like to know if double mutation mice, does also inhibit KIT or not?
Yes, it does. Yeah, I didn't present those data, yes.
Right. So that means the inhibitor goes, why they are sensitive to sunitinib skills go to like KIT?
Because sunitinib still fits into this active site. So even when it is mutated, there has been done modeling based on these structures that sunitinib has a different conformation and not like imatinib can still fit somehow into this active site of KIT and can inhibit KIT